We are interested in the causal interactions measured within multidimensional neural data from two or more electrodes recorded at the same time. This right here is a simplified theoretical model of two presynaptic neurons, 1 and 2, projecting onto postsynaptic neuron 3. With most of the currently available causal analyses, typically what we do is break up a system, such as neuron 1 onto neuron 3, investigate those causal interactions, and then do the same for the remaining half of the system. What most systems struggle with is to investigate the combined effect, the synergistic effect of neurons 1 and 2 onto neuron 3. In a recent publication in Nature Neuroscience, Giulio Tononi's laboratory called these composite mechanisms that are irreducible to their elementary constituents. And the same laboratory has been proposing an analysis path to deal and to quantify with those kind of interactions. This kind of analysis stems from integrated information theory. And the starting point is to create a transition probability matrix, as shown right here to the right. So if we take two input elements, A and B, and look at its effect on element C, what we can do is we can investigate each of the possible states, in this case, A and B being inactive, and then investigate the effect of that onto element C. In this case, you can see that there's a 90% probability for element C to be inactive following the inactivity of elements A and B. Now, for a complete transition probability matrix, what you have to do is investigate all possible permutations of A active, B inactive, and so on, and then find out the probabilistic outcome onto element C. And at the end, you result with a complete Markovian description of the entire system. One way to think about that is that you have a cause as a current state, and then an effect as the next state. But this doesn't guarantee causal interactions. For that to be quantified, integrated information theory adds a second step, where the system gets cut into constituent elements, and then there's a quantitative comparison between before and after the cut. If there's any difference, causal non-reductionism can be assumed. What does this mean in practice? So here is a very simplified transition probability matrix of just two elements. They can either be off, one on, and so on, and then the outcome on a third element. Through statistical noising or marginalizing, we can now cut or eliminate one of these elements and then investigate the outcome onto the transition probability matrix. What we find that in this case is that the transition probability matrix changes after we statistically noise or marginalize one of these elements, therefore showing that there has been a causal relationship and allowing us, by quantitatively comparing between the prior and post transition probability matrix, the size of the causal interaction. Now, earlier this year, Tsukiya's laboratory published an extensive paper showing how to use this approach onto neural data. In this case, local field potentials that were recorded with linear electrode arrays in the mushroom body of a fruit fly. Today, we will show how this can be used for linear electrode recordings in macaque monkeys. This right here is a standard population spiking response multi-unit activity to a visual stimulus in primary visual cortex. And this is the simultaneously collected local field potential, the evoked potential. Now, typically what we do in my laboratory is we record those across the entire depth of the cortical manifold. So from the pia all the way down to the white matter across all layers of cortex. The first problem that arises using those kind of data for these analyses is that we have to eliminate the initial transient. And the way that we did this is by computing an average evoked response and subtracting that from each trial, shown right here, to achieve a pre-whitened or flattened response. Then we binarize the data by taking the average activity in each trial and going into basically transforming the data into up and down states, shown here in white and black, as a function of time. And this can be done for local field potentials or Cohen's density data as well. To simplify, what we'll do here is we'll focus on just three of the main lamina compartments, A, B, and C, with B being the input layer, the middle layer, granular layers of cortex layer for C, and A being the upper and lower layers of cortex. Using this integrated information theory, approach, we can now look at the transition probability matrix that arises from these data, shown right here. There's lots to already infer, such as that the system has a certain stickiness to it and a certain memory resulting into its own state. But to make it even easier to see, we transform this kind of matrix into a graph using graph theory. What I'm showing next here is neural data trial averaged of an animal seeing a visual stimulus right here, and then in blue paying attention or in red not paying attention. And at 4 discrete points in time, I've been computing this transition probability matrix shown here in graph form. And just to summarize, one of the measures that can be extracted from these, which basically corresponds to the amount of interactivity or causal interactions within the system, you can see that it increases the moment the stimulus appears on the screen and further increases once the animal pays attention. But we're not stopping right there. Integrated information theory has recently been extended to version 4.0. And in that case, you can look at the reducibility of the causal interactions themselves. This right here is from a paper from Andrew Hahn and Giulia Tononi, where they constructed a very simple 8-element system. This is the transition probability matrix of that system. And here you can see what they call relations, which is a multidimensional abstract structure that shows all of the possible non-reducible causal relationships in the data. Now, doing this for our data, we were encouraged to find out that only a small number of all the possible causal interactions in the data actually survive, basically giving us a multidimensionality reduction within the causal data itself.